Hello everyone and welcome to Brain Blitz Audios. Today we're sharing some exciting news from the world of science in our series titled SciBrief. So in this particular series we'll be looking at news from the scientific world, and we analyze what makes the news interesting and exciting. So let's start off with today's news, which is BioEmu AI reveals protein choreography in biological conditions. So let's look at the story. So BioEmu AI maps protein dynamics in native biological settings. Now let's look at some background. Proteins are not static structures. They bend, twist, and occasionally unfold. Dynamic movements that are crucial to understanding their function. Some, like enzymes, open like clamshells to capture molecules, while others, such as signaling proteins, alter their shape to regulate cellular processes. Still, some proteins transiently expose hidden pockets where drugs can bind. Although artificial intelligence or AI tools like AlphaFold have made predicting protein structures more routine, they typically provide only a single stable conformation, just one frame in what is in reality a constantly shifting molecular movie. Now let's look at the new tool. A newly developed deep learning system called BioEmu, created through a collaboration between Microsoft, Rice University in the United States, and Freie Universität in Germany, predicts the complete range of shapes that a protein naturally adopts under biological conditions. This range, known as the equilibrium ensemble, enables high-resolution modeling of protein flexibility at scale, something that traditional, slower methods struggle to achieve. As described in the journal Science, BioEmu offers a faster and more cost-effective solution, making it possible to perform large-scale predictions of protein function. Now let's see how this tool works. To grasp BioEmu's importance, it's useful to understand its competition. The gold standard for modeling protein flexibility is molecular dynamics, or MD, which simulates atomic movements at femtosecond resolution using tools like GROMACS or Anton. While accurate, MD is extremely slow and expensive. Simulating microseconds of motion can consume tens of thousands of GPU hours, even on supercomputers. BioEmu overcomes this with an AI diffusion model. Researchers trained it using millions of AlphaFold predicted structures, 200 milliseconds of MD simulations across thousands of proteins, and data from 500,000 mutant sequences based on experimental stability. Like watching a sugar cube dissolve, BioEmu learns to reverse the process from noise back to structure, and can then generate thousands of realistic protein conformations from scratch. Now let's look at some of the statistics behind BioEmu. BioEmu outperformed traditional methods, capturing major enzyme shape changes, local unfolding that toggles activity, and temporary drug binding pockets like those in the cancer-related protein RAS. It predicted 83% of large shifts and 70 to 81% of smaller ones, including open and closed forms of the enzyme adenylate kinase. It also handled flexible proteins without fixed 3D structures and predicted mutation effects on stability. According to Kalaidasan Ponnuswamy, a bioinformatician and assistant professor at SRM Institute of Science and Technology, MD simulates the full motion of proteins and drug interactions, while BioEmu quickly produces thousands of stable shapes within minutes on a single GPU. BioEmu shows the end states of proteins, but not the step-by-step journey, he explains. Now let's look at the next page that builds on top of that. While BioEmu excels at rapidly predicting stable protein shapes, it has clear limitations compared to MD. MD can capture temperature changes, membranes, and complex dynamics that BioEmu's static predictions can't yet model. BioEmu also can't simulate cell walls, drug molecules, pH changes, or provide reliability metrics like AlphaFold. It's restricted to single chains and cannot represent protein-protein interactions, which are vital for biological functions and drug targeting. According to Ponnuswamy, BioEmu should be seen as a hypothesis-generating tool rather than a definitive source, as it advances to handle more complex proteins and chemical environments. Researchers will still depend on experiments or molecular dynamics to validate its predictions. The conceptual leap is evident. If AlphaFold outlined the blueprint of proteins, BioEmu maps their choreography. By rapidly capturing flexibility across thousands of proteins, it enables large-scale drug discovery and functional studies with far fewer resources. Tasks that once took weeks can now be done in hours. However, this progress demands new expertise. Future scientists will need not just strong foundations in physics and chemistry, but also fluency in machine learning and physical modeling to fully harness these hybrid methods. Researchers view BioEmu and MD as complementary tools. BioEmu generates a broad set of plausible conformations, while molecular dynamics explores them in detail. This combination promises faster simulations without sacrificing accuracy. And that concludes today's episode of SciBrief. We hope you found this episode interesting and informative. For more such content, don't forget to subscribe to our channel, which is Brain Blitz Audios. You can also hit the notifications button present below the video in order to get updates about our latest content, whether it be related to SciBrief or our other series. Now, until the next webisode, take care, stay safe, ta-ta for now.